Greetings of the day to everyone. My name is Rishabh Kabra, along with my associates Upasana Anant and Ritish Kamboj, from 21 Integrated MSc Biotechnology at V.I.T. Bhopal. On behalf of my team, we would like to welcome you all, and it's our honor to deliver the explanation in front of you on a very interesting and fascinating topic, that is erythrocyte sedimentation rate, in short E.S.R. So, this is our team. Anurag Kumar MSI, 3. Deepa Yadav MSI, 14. Chirag Sharma MSI, 16. Rishabh Kabra MSI, 17. And next are Ritish Kamboj MSI, 15. Shivani Soni MSI, 10. Shikar Mishra MSI, 6. Upasana Anant MSI, 11. Now let's see what topics that we are going to discuss in this video. First is, talking about the blood, we will discuss blood as a transport system. Second, we will discuss about the sedimentation of blood. Third, is the components of blood. Then, we will move to the E.S.R, that is, erythrocyte sedimentation rate. We will get to know what is E.S.R, how E.S.R works in general, why E.S.R is really crucial. So let's begin with it. Blood as a transport system. Blood is the primary transport medium that is responsible for continuously supplying nutrients and oxygen to the active cells in the body, and their primary function is to transport oxygen and, to a lesser extent, carbon dioxide, also, along with delivering platelets and homeostasis. The next is components of blood. Blood is a specialized body fluid. An average adult is having around 5 liters of blood circulating in his body. It comprises of plasma, red blood cells, white blood cells, and platelets. There are many cellular structures in the composition of blood. When a blood sample is putted in a centrifugation machine, they separate the following into plasma, buffy coat, and electrolytes. Plasma. The liquid state of blood is considered to be plasma, as it makes a 55% of blood and it is yellow in color. Also, it consists of salt, nutrients, water, and enzymes. Moreover, blood plasma transfusion is given to patients with liver failure and life-threatening injuries. Red blood cell. Red blood cells, commonly called as erythrocytes, consist of hemoglobin. They are formed by the bone marrow to primarily carry oxygen toward the cells and carbon dioxide away from it. White blood cells. White blood cells are originated from the bone marrow, and they circulate throughout the body, responsible for fighting foreign pathogens like bacteria, viruses, and fungi. Further classification of white blood cells includes lymphocytes, monocytes, eosinophils, basophils, neutrophils, platelets. Platelets, also known as thrombocytes, are tiny disc-shaped cells that help to regulate blood flow when any part of the tissue is damaged, thereby helping in fast recovery through clotting of blood. And the last is blood sedimentation. Plasma is lightest of them all, as shown in the figure. It remains on the top, while other components, i.e. cells, are at the bottom. To be specific, RBCs are the heaviest in all of them. Plasma composes 55% of blood, and rest 45% composition is of blood cells, which includes white and red blood cells. Blood contains cells, proteins, and sugars. If a test tube of blood is left to stand for half an hour, the blood separates into 3 layers as the denser components sink to the bottom of the tube and fluid remains at the top. The straw-colored fluid that forms the top layer is called plasma and forms about 60% of blood. The middle white layer is composed of white blood cells, WBCs, and platelets, and the bottom red layer is the red blood cells, RBCs. These bottom 2 layers of cells form about 40% of the blood. Let's continue further with Upasana.
Having read about the basics of blood, now let's hop to the main topic, erythrocyte sedimentation rate. Do you know the meaning of sedimentation? It's quite simple. It is the process where particles in a suspension separate out from the liquid and settle down due to gravity. Blood being suspension itself shows this property too, that is sedimentation occurs in blood when an anticoagulant like heparin is added, then erythrocytes settle towards the bottom of the test tube. The fact is, the fastest settling particles are larger, heavier, spherical molecules because the sphericity of sedimenting particles is an important condition for good sedimentation. And the slowest settling particles, which sometimes cannot be settled accurately or properly, are tinier, lighter, and/or irregularly shaped molecules. How do we calculate E.S.R? It is quite simple, and it varies with gender. Like for males it is age divided by 2, whereas for females it is age plus 10 divided by 2. How E.S.R functions. E.S.R basically a blood test that is carried to know the well-being of the blood system. An E.S.R can detect any disturbances in body like inflammation, etc. Why blood is chosen? As already told, blood is a suspension which is heterogeneous in nature. Erythrocytes are the main insoluble solute present, and it is these cells that perform the sedimentation, and the time duration they take to settle is called E.S.R rate. Important points. Different types of particle react different in this test. The action varies with the shape, size, and weight of the particles. For example, larger, heavier, and spherical molecules tend to settle faster and lighter. Tinier and irregularly shaped particles tend to settle very slowly or do not settle at all. Sphericity is an important condition which help in good and faster sedimentation. We went through the fundamentals of E.S.R, and now let's see how E.S.R functions with the help of an experiment. These are the things needed for the experiment: vegetable, tomato, petroleum jelly, vegetable oil, mortar and pestle, a glass container, distilled water, and a knife. The procedure is quite easy to understand. We will wash and cut the vegetables, then we will be mashing it by adding a little water. Then we will mix the mashed tomato with other ingredients, that is vegetable oil, petroleum jelly, in a bowl. Now let's relate this simulated protocols with the real company components. The fibrous tomato acts as erythrocytes, the vegetable oil can be related to plasma, petroleum jelly mimics the role of fibrinogen. We can also add other ingredients like butter that acts like globulin of blood, starch solution acting as WBCs. We can also create diseased conditions artificially. Example, using beet extract a reduced protein condition is reflected, and using beet shavings sickle cell condition will be formed. In order to perform the experiment successfully, it is very important to use clean containers, mortar, pestle, etc. Then we will transfer the contents into a glass container and wait for 60 minutes. Let's watch a video for a better understanding. Over to the calculation we get. After sedimentation, the measurement where tomato smash settle down is 2 centimeters. Time taken to settle down is 60 minutes. Rate of sedimentation equals distance traveled by tomato smash by time taken is 5 into 10 to the power of -6. For results we get: the vegetable gets settled down to the bottom. The oil settles above the oil. And petroleum jelly stays on top. There are 3 levels. In the first 10 minutes, RBC stalk over each other, called aggregation, a slow step. In the next 40 minutes. In the next 40 minutes, the aggregated erythrocytes settle down rapidly. In the E.S.R test, fluid is plasma factor. The particle factor is an erythrocyte factor. In the third and the final stage, lasting about 10 minutes, packing occurs is a very slow step. Now, Ritish will explain further.
So now we will understand why E.S.R is crucial. Because the numerical value of the E.S.R is altered by changes in blood composition, it is of tremendous clinical importance. Because disease alters blood composition, the E.S.R value of blood from a sick individual differs from the E.S.R value of normal blood. So now let us look upon some disease. They are mainly inflammation, infection, cancer, anemia, autoimmune disorders such as temporal arteritis, polymyalgia, rheumatoid arthritis. Now let us look at a specific one. That is sickle cell anemia. Sickle cell anemia is an inherited disease in which a person's red blood cells are disc-shaped, like donuts with no holes. We expect the E.S.R of sickle cell anemia blood to be lower than the normal value because the sickle cells will not settle quickly and tend to stay in the plasma. Let's get to the interpretation. The first is, an E.S.R does not significantly diagnose any disease, but it can provide information that whether or not there is any inflammation in the body. Next is, sometimes an E.S.R may be slower than normal. The slower E.S.R indicates blood disorder. Third one is, the fastest settling particles are larger, heavier, spherical molecules because the sphericity of sedimenting particles is an important condition for good sedimentation. And the last is, the slowest settling particles, which sometimes cannot be settled accurately or properly, are tinier, lighter, and/or irregularly shaped molecules. By winding up the topic, I would like to thank Dr. Neeta Kulra for her guidance and teaching this topic in such an interesting manner that we have enjoyed a lot while working on this. Dhanyawad.